Good morning, everybody. Thanks for sticking around for the last day. I'm really excited to be here. Thanks for the organizers for inviting me. I want to give everybody kind of a heads up that I'm not entirely sure how this is going to fit with everything else we've seen in this program, but hopefully I can convince you that there is some interesting materials properties of materials made of bees and made by bees. Just a little bit of background. My training is in physics and material science. My specialty was on soft condensed matter for my PhD. And as it turns out, and as I found out during my postdoc training at the lab of Mahadevan at Harvard, there's a really beautiful range of interesting materials that bees create. For those of you who don't know, bees have this really interesting social hierarchical structure. There's one queen, there's lots of workers, tens of thousands of workers, and they tend to stay coherently aggregated around the queen, sometimes in these kind of structures that they form for, in part, thermoregulation purposes when they migrate to a new nest structure. They use really similar structures as a scaffold when they're building their honeycomb. And when they're building the honeycomb, they're also creating materials in a sense. They're making these little wax blobs, and they are somehow assembling them into these really beautiful three-dimensional hexagonal lattices where they store honey and other resources. So what I want to talk about today is a couple of these processes, but I'll really try to put this in material science terms. So the first part I'll talk about the self-assembly processes in which honeybees aggregate into a dense group. Then I'll talk about some of the mechanical aspects of the structures that they create, especially when they become three-dimensional. And then I'm going to switch to talk about switch from materials made by bees. The building blocks are the bees in these kind of structures, to manipulation of bees of inanimate materials like wax and how they create these honeycombs. So feel free to stop me for questions, if anything, if biology or anything else. I'm happy to take questions in between. So I'm going to focus on, well, I will say more generally the goal in my lab is to try to understand this biological behavior and identify the rules of behavior in which these individuals follow in order to achieve mechanical stability, aggregation, distributed construction, and so on. We use behavioral experiments. We use mathematical and computational modeling to show as a proof of concept that some of these ideas might actually work, at least in simulations. But we don't really fabricate materials. And my really personal dream is that maybe some of these algorithms would inspire some of the amazing experts we have here in the audience to maybe fabricate something based on these rules. So let me start with self-assembly. Okay, so this is a movie showing a commercial honeybee hive. This is this little box here. All these bees, these are the worker bees, and they somehow know where to go. The queen is already inside. And I'll play this again, they sort of march, they march up into the entrance of the hive, which is this really, really narrow slit over here. And how do they do this? It turns out if we look very closely at the entrance, this is what we see. So hopefully you can see a lot of bees kind of fanning their wings. And what they are actually doing is producing a chemical signal in which they use to communicate to other bees and direct them towards the entrance of the hive. So over here, there's a little pheromone gland, and then they fan their wings. So they create airflow across that pheromone gland and potentially moving that information or pheromones backwards towards maybe less informed bees. And what we noticed, this is especially the project of Dumai, who just graduated from my lab this summer, we noticed that bees would stay at a particular distance from the entrance and start producing these chemicals. This is a behavior called scenting, by the way, because they produce scent. So let's say this bee is starting to scent, and then another bee would stop at a particular distance and do the same thing, and then other bees and other bees and other bees would do that. So they create this really interesting communication network that eventually gets them to aggregate around the queen. So we wanted to study this a little bit better, and we started with a set of experiments where we had the queen confined in a semi-two-dimensional arena. The rest of the bees were also in the semi-two-dimensional arena, so there's no flying. We're just simplifying the system into a two-dimensional system. And in order to classify the behavior of the bees and identify these scenting behaviors, we collaborated with Greg Stevens and created these nice machine learning classifier for behavior, which basically tells us is this bee scenting or not scenting, and if it's scenting, in which direction it's producing that signal. Okay, so here's a typical experiment. We're looking at the arena from the top, from an aerial view. Here's the queen. The bees are going to start over here. Every time you see one of these blue arrows, that's when a bee is scenting and producing a communication signal. And I'll play this movie. This is a little bit sped up, but as you can see, there's lots of scenting behavior, lots of communication, lots of blue arrows. And if I scroll a little bit further, if I can, at the end of the experiment, they do find the way to the queen and they aggregate around her. So there's a few, this is really just kind of a proof of concept that the scenting is associated with that aggregation behavior, but there's a few other things we can learn from these experiments. In particular, since we know the directions of the communication spots and the location and direction, we can treat them as little gradients and integrate them into a higher dimensional surface, where if indeed all of these communication signals integrate into a global map that leads to the queen, we would expect the higher values of the surface to correspond with where the queen is. And here is an example from one experiment. There's more of this in the published paper. The other thing that was kind of interesting is that we noticed that the bees who produce signals distance themselves or position themselves at a very particular distance from each other. And if we think for a second about diffusion from a source, we know that a particular distance from a source would correspond to a particular concentration. So potentially, it could be that the bees are really being triggered by a concentration threshold that gets them to start producing the signal themselves and also potentially follow these gradients. So this is as far as the inspiration from the experiments go. And then to try to see if that could explain the aggregation behavior, we created a really simple agent-based simulations where the agents or spheres or bees are now just moving in a two-dimensional environment. They tend to move in a random direction until they bump into a cell that has a higher a concentration of signal that is above a certain threshold. And then if they do that, then 50% of the time they start producing a directed walk up the pheromone gradients, potentially towards the individual sources or the queen. And then the other 50%, they produce their own signals by scenting. And for this, to produce this, we really just solved the classical diffusion equation with a directional bias. So this is, of course, has been done a lot for biological systems. There's lots of examples of biological systems that produce and also sense the same chemicals to aggregate, like social amoeba, slime mold, some of you may be familiar with. But I think really the interesting trick here is that the bees have the directionality of the signal that they produce with airflow, and we really try to capture that in our simulation. So when we run the simulation, we have the luxury of changing some of the behavioral parameters that maybe we wouldn't be able to ask the bees to change. For example, change the directional bias, where 0 means that it's an isotropic signal, and high value means that it's very directed. Same thing for the concentration threshold, so low means that they get triggered more often than high values. And we ran this parameter sweep to create a phase diagram where we're seeing a few different behaviors. So for low thresholds and high directional bias, then the queen is here in the center, but none of the bees really get triggered because the concentration threshold is so low. At the opposite side of the phase diagram, we have a state which is analogous to local equilibrium, where many of the bees get stuck in these small little clusters. Presumably after a long period of time or with additional noise of temperature, they might be able to find their way to the queen. Another phase, which is diffusion limited aggregation. So all these bees over here are going to randomly walk in the arena until they bump into the big cluster in the middle. And of course, that scales very badly with system size. And then there is this sweet spot with clustering that happens when the bees are able to actually propagate that signal forward or backwards to all the bees in the arena, and they're able to aggregate coherently around the queen. And for those who are familiar with aggregation processes, and I'm assuming there's a bunch of you here, none of these phases are really surprising, but I think what's really interesting here is that this more optimal phase or optimal aggregation happens in the presence of directional bias, which hints that directional bias is making this whole process a little bit more robust. Okay, and we also try to make the bees' lives harder by adding some obstacles, like we get them to solve these mazes, or this is a classical Y maze, and we were actually able to show that the bees not only find the way to the queen, they also find the shortest path to the queen, which has some interesting cognitive implications for the bees. All of this is in 2D, but actually when the bees aggregate and also in their day-to-day lives, inside the hive or outside the hive, when they create these structures, the structures that they create are three-dimensional, and they're actually usually suspended. So this is an example of a honeybee swarm. It's really important to emphasize that there's nothing besides bees and a tree branch here. So the bees are just holding hands in a sense, and this is a material made of bees. And there's 10,000 of individuals in these kind of structures. So of course, there's going to be some mechanical implications to this, as well as the bees need to be able to adapt to environmental perturbations, like the ambient temperature is actually getting the bees to change their density. So here is this is the same swarm at high ambient temperatures and at low ambient temperatures. And when it gets low, they kind of huddle. So they increase their density for thermoregulation purposes. But of course, this is all going to be coupled to the mechanics of that structure. When it rains, the bees they align their body axis along the vertical gravity line. So they create these shingle-like structures that get the swarm to be less susceptible to water. And then at really high temperatures, it's been speculated a long time ago, or a couple of decades ago, that the bees actually create air channels inside the swarm to be able to ventilate their hive. So all of this is complicated, but as I already alluded to, this is going to be coupled to the mechanical stability of the swarm, and all of these processes that the bees do it has to happen while the swarm is mechanically stable and doesn't break. And the bees adapt their shape to the shape of the swarm to the shape of the branch to account for more static forces like gravity, but also more dynamical forces like if there's a windy day, the swarm might get mechanically perturbed, and if the bees break, that can be pretty catastrophic for the survival of that colony. The question that we wanted to answer, and this is actually a long story that started a while ago when I was a postdoc, is how do these bees maintain mechanical stability given the fact that there's so many of them, there's tens of thousands of individual of bees. So a bee at one end of the swarm might be able to communicate with more locally with bees around her in her vicinity, but not with bees at the completely other side of the swarm. And we're trying to understand what kind of local cues the bees might be able to use to solve that problem. So the idea for the experiment was by Maha, who was here, is not here now, but he was here a few days ago. And the idea was kind of simple, but a little bit crazy. We decided to build a swarm in lab conditions and shake it and see what happens. This is how a typical experiment looked like. Very frugal. We had a motor that controlled that plate, and this is a swarm over here. You can probably already see that it's a little bit jiggly, so there's going to be some mechanical and elasticity involved in here. And what we found out is that when we continue shaking the bees right and left, bees inside the swarm move around and they collectively change the shape of the swarm. And this is a sped up version of that experiment. So this is the front view, this is the bottom view, and I'll play this maybe at least twice so you can see what's happening. So we start with a swarm that has a cone-like structure and with a certain base area and height. And as we shake the bees, they kind of spread out like this. So they decrease the height and increase the surface area. So, that was kind of interesting, and we decided to try to understand what kind of mechanical information the bees might be using in order to achieve that response and changing the morphology and the shape of the swarm collectively. Then again, came back to my comfort zone of more soft materials modeling, a very, very simple model of the swarm, but we think it can explain some of the outcome of these experiments. So we have spherical bees connected by elastic springs, and we have some excluded volume interaction so that the bees cannot really overlap. We have gravity, and then we solve the equation of motion that include some friction, and we include friction because we know that when we shake the swarm for a while, it will come back to a steady state and it won't just continue shaking forever. We're looking for the mechanical cue that the bees are using, and we, as a candidate, used the instantaneous deformation or stretching of bonds that bees create to their neighbors. So this is illustrated over here. And then we just take the geometrical average of that, which is shown here in this big equation. But it's really just looking at all the bonds that a single bee is creating to her neighbors and taking the average deformation of these. So here is the simulation. This is a three-dimensional swarm cut in the center. We can compare what happens for an elongated swarm versus a more spread out swarm. And of course, as you might expect, spreading out is lowering effectively the deformation that these bees over here are experiencing. It's a little bit, it's the same principle as if you're standing on a shaky ground, people kind of tend to crunch down. So I think this is the same kind of effect that we're seeing here. And then the model actually made some interesting prediction, which is that at least for this configuration, which is very ideal and probably not realistic of the bees inside the swarm, we should get a really different response when we shake the bees right and left horizontally versus up and down vertically. So the signal here is going to be much lower than the local signal over here of the stretching of the bonds. And as it turns out, we were able to validate this prediction. So what I'm showing you over here, this is the spread out of the base. So this is the base area of the bees. The relative base area of the bees is a function of time. Different colors corresponds to different forces or accelerations that we applied on the board. And the dotted line over here is when we stopped the perturbation, and we can see how the swarm is morphing back to its original shape. So this is the situation where the swarm does respond, and because we're shaking them right and left. However, when we shake them vertically, nothing happens in the sense that they don't really adapt, they don't spread out, they don't change the shape, even when we really cranked up the acceleration. And actually, we try to crank it up more and more, and then at some point, this whole thing really just breaks, which is inconvenient for everybody, the bees and also us, because it takes about a couple of hours to get them to reform again. So we didn't explore this regime too much experimentally, but we were. It was a good point of reference to at least conceptually validate the model that made this prediction. And finally, I just want to tell you that we also used all of this information to come up with a simple extension of the simulation that I've shown you so far, where instead of just passively bees being connected to their neighbors, and these connections in the previous simulation were fixed, over here we're making this a little bit more flexible. So now bees can actually break bonds and use the local signal, the local mechanical signal that we found, which is the stretching of the bonds that they make to their neighbors. So they sense that. We can choose a particular threshold that triggers a behavior where bees would tend to move upwards towards the attachment surface, and this is how it looks like. And I just want to say that so we implemented this based on a local rule. So the bees are really just sensing locally the mechanical stretching or deformation of the bonds around them. And this is the only information that they're using in order to move upwards, but they're actually just moving up the strain gradient of these local deformations. We could also implement this in a more straightforward way, potentially by just programming the bees to move upwards without any local information or directionality. But we were not sure that bees inside a really shaky, dense environment would even know what upward is. So we implemented this based on this local rule. And just to be consistent with the experiments, we chose a threshold that triggers that behavior such that bees in the vertical shaking are not going to get triggered as much, so they're not going to change their shape very much. But of course, if I would ever be able to design such a material and have the luxury of choosing this threshold that triggers the behavior, I would choose it low enough so that also the vertical case would get mechanically stable for more synthetic systems. Okay, so right, need to speed up a little bit. Everything I showed you was from the what we can learn from looking at the shape of the swarm from the outside, looking at the bees on the surface. But if you do a really quick back-of-the-envelope calculation, the majority of the bees are inside the swarm, and we just never were able to see them because of the opacity of the swarm. So fast forward a couple of years forward, I opened up my own lab, and the most expensive piece of equipment that I bought was an X-ray machine, so that we can actually peek into the inside of these swarms and try to understand how bees are organized inside. So this was led by Olga, who's a postdoc in my lab. The swarm was rotating in between the emitter and detector, and we were able to get things like this. So this is basically a three-dimensional reconstruction of the swarm. So we have a 3D map of how bees are arranged in the swarm. And we just published all of this data, and I'll tell you about a bit of analysis that we did, but I think there's hopefully more to come. So we partnered with Kaushik Yarman, who is now faculty also at CU Boulder, and we tried a really, really basic exercise where we are trying to understand how different layers of bees are arranged in order to maintain mechanical stability and in order to support all the bees that are suspended underneath them. And this is basically a strength mass scaling law that I'm sure some of you are familiar with. For humans, there's a or for other single animals, there's a very particular scaling law that we would expect. And as it turns out, for the bees, there's also, the data suggests that there's also a scaling law. So what I'm showing you here is we're taking slices of bees, and we're looking at how many bees are in each one of these slices. Really, really simple calculation. And we're also calculating how many bees are underneath, and this is what is being plotted here. This is the mass of a slice, and this is the weight that it's supporting. All the different colors here correspond to swarms of different sizes. So, and they seem to fall on this universal curve. And I'm gonna probably skip fast through this because I'm running out of time, but I'll just say that we were also able to build a simple continuum model of this arrangement of the bees inside. We're presenting the weight that the bees are supporting and their own weight in the slice as a scaling law. And we can try to understand how the exponent in the scaling law would change that mass distribution. And I'll just put this on here. So the gray area here is the experimental data. The black is the exponent that we get from the experimental data, and then there's all these other really non-realistic situations, of course, that the bees would probably never follow, but we have some analytical understanding of them. Okay, so I think I'll move to the last little section here. So I showed you how bees create materials from their own bodies, how they aggregate and how they create stable structures in 3 dimensions and change their shape to adapt to mechanical perturbations. And I'm going to do a little bit of a leap here to start talking about how bees, as a swarm, manipulate inanimate structure, which is wax. And a little bit about the honeycomb structure that the bees create. So this is it's a really beautiful, pristine structure when it's unperturbed. So it's a 3-dimensional structure based on a hexagonal lattice. The bees, it's really important for the survival of the colony. But it's also kind of expensive to create. So the bees actually have these, they generate the wax. So they have this little 6-pack over here, and they generate these little wax flakes, and they need to consume 8 times more honey to generate that amount of wax. So it's really expensive. So they want to minimize the amount of materials that they create. And as we all know, hexagonal lattice and these kind of structures at least have been known for quite a while to minimize the amount of material while maintaining a higher cell size. Okay, thank you. This is in a perfect world. This is what the bees would do, but actually in reality, the bees need to deal with a lot of geometrical constraints, especially feral swarms that tend to nest in bee cavities. So they have to deal with all this internal structure of the cavity, and of course need to deal with boundaries that might drive them away from this more optimal situation. So we were really interested in understanding how the bees do this. And by we, I mean Golnar, who is a graduate student in my lab, co-advised by Francisco Lopez Jimenez, and also Daisy Zhang. I don't know if she's here, but she was actually working with us as a high school student, and now she's an undergrad here in Princeton. I think she's going to show up maybe a little bit later. So what we did is use a trick that beekeepers use to get the bees to create a particular structure, and it's called a starter frame. So you see it over here in blue. So this is a This particular one is 3D printed. We give the bees this little starting pattern, and to some extent, or in most of the time, they adopt it and start building their honeycomb, and it's mimicking, or at least starting to mimic the pattern that we provide them. So we want to understand how bees deal with geometrical frustration. The idea was, why don't we create impossible problems for them to solve? So we designed 3D printed lattices that had two lattices, two fronts, with a gap in between. And the two lattices were just a little bit off, so there was an angle problem, mismatch, or there was a horizontal and vertical shift. And we give these to the bees. Here is a starter frame, and then the bees adopt it. And then from this, we can learn something about how they deal with this geometrical frustration. So as it turns out, I'm going to show more data, but this is really just one particular example of the interface. So this is where these two lattices front met. And by doing computer vision analysis of these structures, we can find defects along the interface line. So by defects, I mean that instead of having 6-sided cells, the bees create 5-sided or 7-sided cells. And they seem to have a really particular structure. You can see that there's a connecting line here that connects defects of 5-sided 5, 7, 5, 7, 5, 7. And as it turns out, and maybe some of you already kind of recognize this is really similar result to what we would expect for a simple crystal structure simulation with Lennard-Jones interactions. And here is an example of that simulation. So what we simulate is the centers of the cells, and they can move around and interact with each other using a Lennard-Jones potential. And here we just ran a Monte Carlo simulation where we try to minimize the energy. So the centers of the cells are moving around. They're starting from this really energetically costly configuration, and then it converges on something similar to what we're seeing with the experiments. So we decided to study this a little bit more systematically. Here again, this is the comparing the experiments with the simulation of two lattices where they have an angle mismatch of 30 degrees, which is the highest mismatch we found. But we can also explore other angles. So again, each panel here is showing both the experiments, which are the gray area, the gray cells over here with the simulation. And it seems like the simulation, this really, really simple simulation, can make predictions that the bees follow for different angles. In particular, when the angle mismatch decreases, we can see that the distance between the pairs of defects is increasing. This is, of course, just one simulation and one experiment, but we also did this more systematically for a group of these experiments, and you can see some of that in our paper. It actually just came out 2 weeks ago. Okay, we can also see the effect of vertical shift. So here in particular, the simulation would predict to see pairs of defects that have a very particular angle. And we can see that both in the simulation and experiments. And of course, we also have the control where the lattices are actually perfectly matching. And so there's no defects both for the simulation and experiments. So I feel pretty good about this model being able to explain two-dimensional pattern formation in these kind of situations in honeycomb formation. And by two-dimensional, I mean that the cells are actually just bulging in a straight angle away from the frame. But, and here is a little bit of unpublished data, but I'm very excited about it and wondering what you guys think. There is a lot of three-dimensionality to that structures as well. And we started just for fun to, we had the angle mismatch, the vertical shift. We also wanted to test what happens when we give these cells of sizes that they are maybe less familiar with. So we just changed the cell size. It's another way to manipulate the structure and create geometrical frustration. And here we see something a little bit different. So let me orient you. We give the bees cells. These are the cells that they're kind of the cell size that they're used to. So they completely adapt that structure and they create vertical walls. By the way, this is honey. This white spot over here. But then as we give them cells of more and more increasing cell size, they start tilting the walls. And to be able to see that, we actually need to use X-ray to measure these angles. So this is the X-ray 3D reconstruction of that frame. This is the plastic, and the bees create some stuff above it. So we think that they do this to prevent the honey from leaking out. So if anybody has ideas of how to use things like the meniscus shape, or to try to better understand how they choose that angle, that would be really cool. And the last thing I want to show you, and this was really mind-boggling for us, I have zero idea what to do with this, super happy to get suggestions, is when we give them even larger cells. So tilting is not going to solve that problem. And what the bees do is this. This is the top view. And what's happening is that the bees create an additional layer of wax. And it took me a while to understand that structure. So I think it's maybe a little bit clear when you see the 3D rendition. So they create a second level of a honeycomb. But it's really aligned with the original lattice. And of course, the models that we have so far with the simple, Lennard-Jones particles is probably not going to be able to explain that. But if anybody has ideas, I'm happy to discuss. I'm here till the end of the day. Okay, so I'm going to close up here, skip the summary. Just really want to thank all the people who worked on this, from high school students to undergrads, graduate students, beekeepers, some older and newer colleagues and funding sources. And that's it. Thanks so much for listening.
Thank you. Very interesting talk. We have time for questions. Yes, Americo.
Excellent presentation. My curiosity is, you showed Lennard-Jones potentials in two different situations there, apparently explaining the local interaction and then the pattern formation of the honeycomb. Why the bees choose this law, this potential?
Yeah, and I should say, I think we choose Lennard-Jones interaction because it's a potential that we all know and love. And we know how it behaves, but it doesn't have to be these particular exponents. And I think we can probably reproduce a really similar behavior with a hard wall cut off of the repulsive part, for example. Yeah, there's probably no reason.
You have an idea of the sensibility of these exponents. If you change a lot, go to a certain range, that this can change drastically the behavior?
No, we don't.
Okay. Sure.
So you showed us the honeycomb structure. I saw another talk from at the conference. They said its origin is not honeycomb structure. It could be square lattices, could be triangular lattice, and then they adapt. Final shape is honeycomb. So I wonder whether you have looked into this.
So I don't know if this is what you're referring to, but the cells that they start when you look from the top, they're more square. They're more round. Actually, they're almost spheres. Right. And there's some very active debate about how they move from that rounded shape to the hexagonal shape. We do have temporal data here because we take snapshots of the comb every day or so. So we might have the data to explain it, but we haven't looked at it, and it could be really interesting.
So I was wondering, so eventually it's rigid as a honeycomb, but originally when they produce the honey, it's soft, right? So that takes some time to gradually adapt the shape and eventually forming a solid honeycomb, is that right?
Yeah, so I'm not sure the honey has a particular role in creating the hexagonal shapes. We've seen them do this in the absence of honey. The honey comes in when they deposit it after the cells are constructed, as far as I know.
Okay.
Yeah.
Take two quick questions, and then we, if you can go ahead, and then you.
Yeah, the big eye.
Talk. I loved it. I'm so excited. On the first, on the first topic that you were discussing with the pheromone and they're waving their wings, there must be an energy cost for each bee to be doing this, right? So how does a bee decide that it's going to do that? Like, what's the do you have any idea on what's going on to tell a bee like, oh, I should be doing this. I'm close to the cluster, and I need to tell my other bees to come.
Right. So this is a question I think about a lot, and it seems to be a little bit stochastic. So the number of bees that would be sending, producing that signal, is proportional to the number of bees we have in the experimental arena. But so far, we were not able to really track an individual bee and follow just this one, time series and how she made decisions. And this is something we're working on now with slightly better computer vision tracking, and I have a feeling that these longer time series might help answer that question, but I'm not sure.
Go ahead.
I had a question about the last part you showed with the bees changing the angle
Mhm.
of the honeycombs. Is that like growing on a horizontal surface or
Yes.
vertical? And have you seen?
Oh, no, not a horizontal. So this is a cross section of this slice over here. So in reality, it looks like this right now, but it's actually oriented like this. Does that
And
make
do
sense?
you look at all of it if you change the
Oh,
orientation?
if we change the orientation, oh my god. Yes, great. And it's fairly easy to do with the bees. Good idea.
Final, Grace, question, and then we final question.
Okay, so for the honeycomb, it's clear that like the densest packing of bees is compatible with sort of the smallest perimeter tessellation of the plane, right? That's how you get to honeycomb. But it's not obvious to me if that's true for these gap structures. Do you know? Are those shapes? So you're using this like Leonard Jones kind of like densest bee packing model, but are those also sort of the optimal smallest perimeter cells that you would have, or is this a place where kind of the two models break down, where they differ? Does that make sense as a question? Maybe this is the worst question to end on, but
No. So are you asking about, are the bees still optimizing the cell size?
well,
given
yeah.
these
Do they
defects?
happen to be optimizing the perimeter? They're trying to minimize how much wax they have to produce, right?
Right.
Yeah, so do those shapes also minimize the perimeter, those funny defected configurations?
Versus what situation?
I don't know, versus...
Because we know the optimal situation would be a perfect hexagonal lattice.
Yeah. So if you tried to minimize the perimeter, given those
Okay.
constraints
Mhm.
that you put on, would you get similar shapes? Or is this a place where it's clear that the bees are packing themselves and not actually minimizing the wax?
It's a good question that I don't think I know the answer to. I think we'll need to look more carefully at the wax content there. But I can tell you that the cells areas are definitely not optimized. They compromise for the cell area, and that would probably have an effect on the wax content as well.
Okay, let's thank our speaker again.